everybody to the Physical Genomics um, webinar. Very happy to have such good attendance today. And it is a really great pleasure for me to introduce our speaker today. Dr. Luay Almassalha received his PhD at Northwestern University, and then he went on to receive his MD from the Feinberg School of Medicine, also at Northwestern. And currently he is a 6-year fellow in the Department of Gastroenterology and Hepatology at Northwestern Medical School. He is a Starles Research Scholar, and I believe about to join the ranks of faculty at Northwestern University as well. I personally find Luay's research incredibly innovative and transformative. I've been a big fan of what Luay has been working on. I think he seamlessly combines computational genomics and genome engineering, mechanistic studies, physical biology of the genome, all the things that together comprise this interdisciplinary direction of physical genomics. I find your research beautiful, and I'm just really listening what you have to say. So without further ado, I would give you the podium.
Thanks, Vadim. That's very sweet. And I want to just say, like, obviously this has been an amazing group effort. I have you and Igal as mentors to thank, and across the entire lab groups, it's been an effort over almost 15.5 years to kind of get to this point where we can start to probe how the physical organization of the genome is impacting the behavior of tissues in my patient clinic. So I think this is going to be a wonderful thing going forward, and I hope everyone's excited to hear this talk. And I want to just give credit to everybody, mentors included, for this talk. There have been a lot of wonderful collaborators who made this possible. I'm going to highlight some of the work with specific collaborators on this talk, and some of them have guided some of the information that went into this, but the papers with them weren't necessarily included. And then a lot of different funding agencies have supported this. So thank you for excellent collaborators, mentors, co-workers, and the NIH and private foundations for supporting this. I am a physician scientist, so I constantly think about medicine from the perspective of what I see in the lab, and the lab constantly about what I see in clinical practice. And the most central question for me is, how can we make a system that works across our entire lifespan? We have humans that can live up to 116 years old, and we need to have a model that accounts for all of the length scales of their tissue, from the DNA structure all the way to their human aging. And we tend to think about the process of learning and development in our cells, learning their structure from the lens of development. So when you look at development, there are very tightly coordinated transformations in the genome that happen as a function of time. And that coordination involves activation and suppression of different elements of the genome, depositing different chromatin marks on promoters, the part of the gene that turns things on. That's kind of how it's easy to think about, but it gets more complicated when you start to think about it in space. And it's important to consider that we don't just stop developing and stop learning when we're done as babies. And so this is a very nice photo of my daughter over a year. You can see the rapid transformation, and you see this in everything from mice to cats to humans. And so we need a system that accounts for this. And that system has to work in dividing cells and non-dividing cells. Now, the elegant paper from last year, published in Science, showed that the capacity of mice to form neural networks to solve problems was actually encoded in the organization of the genome. And so non-dividing cells have a system that uses the genome to solve problems. And so where is that encoded? How is that structure, maintained, and how does it integrate across our entire lifespan? There are two huge problems to this. The nucleosome remodeling enzymes modify nucleosomes. They're not guided by specific sequences. And how can I have something that is dynamic? How can I have something that takes our prior states and then integrates that information into a new state all across the tissue? We should think about the context of all of this. So the esophagus, which is part of the GI tract, so part of my world, it's a 20-centimeter long tube. It's made out of a lot of different cell types. So we have to account for the cell types that are present there. And then we can start to think about finer length scales, things that are on the size of chromosomes, how they connect in space, and then how chromatin in those connections is then modified, and then how specific genes, which are less than a few nanometers in size, are modified. Integrating with all of those measurements of the molecular state of the genome, the cell, and the tissue are actual physical structures. So the esophagus is a static organ. It constantly contracts to move food down. A cell is not just a storage of information, it's a physical object. It integrates the molecular state with the behavior of its physical structure. And then that structure extends from micron scale down to the nanoscale, down to individual nucleosomes. And so I'm going to talk to you today about how to solve that problem. And that problem is in some ways like asking, why is my cat in my apartment in a planet the size of the Earth? And is the structure of the Earth being modified by, the cats that are present? And that's kind of an outlandish proposition, but that's the length scales of the problem that we're dealing with. Our cells constantly integrate information about mechanics, electrostatics, and molecular cues. And so on the far left, you can see a contracting nuclei on glass. You can see the volumes are changing. The local density at the nanoscale is changing over time. And this is happening in some tissues as fast as 1 per second. Concurrently, our nucleosomes have to deal with a lot of charge. The proteins themselves don't have enough positive charge to deal with the negative charges that are present on the DNA. Simultaneously, when you get new signals, when you are sick or there's inflammation going on or cancer starting to develop, all of these signals are interacting with the constant evolution of mass and the electrostatics of the system. So we want a system, we want a model and a framework that's going to span all of these considerations. It has to work for the physical components of the genome. It has to work with the chemical components of the genome, so the charges. It has to integrate molecular information. I can't just rely on using stem cells and dividing cells to solve this problem. I have to be able to account for non-dividing cells like muscle, neuron, and cardiac tissue over a whole lifetime. It has to work when we're developing and when we're 100 years old. It has to tell us something about diseases of aging, such as cancer and aging itself, as well as inflammation. And then, like I said, it has to work across 10 to the 7 length scales. Realistically, there's not enough time in a 1-hour talk to do all of that. So I'm going to tell you about how physical genomics can solve that framework. And I'm going to tell you about how our genome uses physical considerations to encode memories, to encode signals. To do that, we first have to talk about how to translate the genome from a one-dimensional object into a 3-dimensional space at the scale that interacts with RNA polymerase. Your RNA polymerase is around 5 nanometers in size, the largest subunit. Nucleosome is around 5 to 10 nanometers. And at the smallest scale, we know it is an irregular network of beads on a string. And then interestingly, it organizes a mass fractal. And what that means is that the larger length scales, the length scales at a domain, are a power law of the underlying structure of the linear chain. And we can capture that with chrome-SEM tomography, and that can provide us with a very powerful set of information about how the genome works. And starting with the 3D structure is really powerful. I, as a parent, watch Inside Out a lot. So many of you may not be familiar with that, but it should be intuitive to understand that if you start with a 3D object, so characters in a movie, and I start to decompress that information into 2 and then 1D information, you can sort of recover the features that map from the 3D object into the 1D object. But it's much harder for me to tell you that you have a pink line and that's equal to an elephant on the far left, and a yellow line is equal to, happiness, which is also an abstract thought, without some additional information. So if we start with the 3D, we can actually go pretty far. Problem with chromatin is we have so much more one-dimensional information than we have three-dimensional information. We have accessibility data, we have RNA data, we have all of the modifications of the genes. We can actually manipulate one-dimensional information quite well. And then we can start to measure things that are connected in space. And I call that 2D information or connectivity information. And then on electron microscopy and on imaging, you can start to capture the 3D volumes and structure of the genome, but that's where we have the least amount of data. And so starting with the techniques that we have through the Center for Physical Genomics and Engineering, why don't we work our way backwards? And that then allows us to say, well, if we know what something looks like in 3D space, can it tell us about the 2-dimensional and the 1-dimensional structure, and then tell us the function that's actually happening? And that's because all of the activities that our cells do happen in 3D. RNA polymerases aren't transcribing necessarily on a straight line. They're transcribing in a space that is well folded and has a relatively irregular structure, but it contains information from that irregularity. And so our group and other groups have shown that at the nanoscale, chromatin organizes into domains, and transcription seems to happen around the outside of those domains. So why does it do that? Now, first question is, can we map features of connections to features of volume? And that's kind of a central question. It's easy to hear the word domain. You are someone who does a lot of research in chromatin and fill in topologically associated domains. So the first question we ask is, are packing domains topologically associated domains? Initially, you would think that this is a solved problem. There was a really beautiful paper by Bintu and al. In Science in 2018, where they used super resolution imaging, multiplex fished, and you can see that the connections in the genome have a spatial distribution. So things that are strongly connected to each other remain strongly connected to each other and are closer. But if you look carefully, some of the sizes on these structures are quite large. So you're looking at 450 nanometers over a maybe 1.5, 2 million base pair segment. And for context, the 240 million base pairs that are in chromosome 2 have a radius of only about 1.5 micrometers in differentiated cells. So it doesn't make sense from a volume perspective. And if you think about connectivity measured by fish, then you can start to say, okay, well, I have these features of connectivity, but I should really ask, are they features of volume? When you go back and you look at the literature about how to do hybridization, there's a key paper out of 2002 that describes the sequential impact of formamide fixation and labeling on the volume structures of chromatin. Now, electron microscopy 24 years ago is very different than electron microscopy today, but it's important to realize what that's showing us is that higher order structures, the geometry and the space filling, is falling apart when we try to dehybridize DNA to label it. And so that may account for that problem. And so we tested that. And so we used our spectroscopic imaging, which is sensitive to nanoscale domains in live cells. And we just sequentially went through the process of fixing cells, denaturing them, and then exposing them to probes. And what we found is when you use formamide, you collapse the structure of domains. And so you see a swelling phenomenon. A structure that maybe was around 70 nanometers is going to occupy a volume of around 450 nanometers. So we're losing geometric information about the space. Super resolution microscopy is potentially a solution to that problem. So there was a beautiful paper in Science Advances by Myron et al. In they went through systematically this idea that chromatin organizes into domains. And when they depleted Rad21, what they observed was that the actual domain structures are largely unchanged. There's no difference in the content of heterochromatin and euchromatin. Their size is about the same, and the surface area to volume ratios are relatively stable. But we need a tool that can span all of the length scales of the genome. It has to be able to go from something that's about the size of a nucleosome or smaller individual strands of DNA all the way up into physical structures. Super resolution imaging with stochastic imaging would naturally be a good tool for that, but it also has limitations. When you are trying to image with super resolution imaging, you need a dye or contrast agent to label the structure. And so we did an experiment where we took EDU and BRDU, which are both conjugates of thymine, and we added them to cells, and we looked to see if the size of the dye was impairing the spatial distribution of the volumes. BRDU requires antibodies, which are around 10 nanometers in size. It's a fairly small object, except from the perspective of the genome. The click-it reaction for EDU needs a 2 nanometer object, and the two together show us something really interesting, which is that a high density area and a low density area, depending on how you probe the structure, can have the same density. It doesn't tell you how things are spatially arranged relative to each other, but you may not be able to recover density and volume information as you would have otherwise expected. That brings us to Chromium. Chromium provides a solution to this problem. So Wu and al. In 2018 demonstrated that you could take a DNA-specific dye, DRAC5, interact with DNA, and then when you photoactivate it, it starts to cross-link with each other, and then you start to be able to deposit in a density-specific way osmium tetroxide specifically to DNA. So now you have a method that has a small molecule dye, it's DNA-specific, and the amount of contrast is proportional to the underlying structure. So we tested the hypothesis. Is a TAD a volumetric structure? Does it have the same properties as the packing domain that we see in space? And we used the same schema that was done by Rao and al. In cell, which was depleted Rad21, and then we measured packing domains. Really interestingly, you don't see a change even at the nanoscale. So down to the structures that are around 50 nanometers, when you look carefully, the density distribution is the same, and the geometry of the domains is the same. We can quantify different properties of packing domains, how dense they are, how big they are, and how they scale in space. So how does the monomer relate to the larger structure? And with and without Rad21, we actually don't see a difference. So the domains are relatively resilient to this process. With respect to the length scales of this question, if we were to rely on just the super resolution imaging modalities, we'd only be capturing the tip of the iceberg. We only capture the largest structures that are present in the nucleus. But Chrome-STEM technology allowed us to ask questions about the entire domain structure and life cycle. So what if we thought about these as dynamically evolving structures that organize the genome? For something like that to happen, you need something that works as an allocator. So you need something that says, take this linear segment, assign it as a unit together. And if it does that, and it does that strongly, you'll start to get a nascent domain, something that's relatively poorly packed, but small. Then you need a system that matures these. So it takes those low density structures, and then it starts to efficiently pack them to deal with the crowded environment of the nucleus. But you don't get one individual structure, you get a distribution of states, and they potentially are moving across each other. And they're not static. So something has to keep them stable. And if they start to fall apart or you need to recycle the material, you have to have a system in place for doing that. And that echoes a life cycle system. So now you have a way to assign a segment of DNA, create a physical structure, and then reuse it as needed. What does that? We did some modeling work the Schleifer Lab Group, the project led by Marcelo Carignano, building off of work by Kai Huang, where we took the concept of force returns. So if you took a polymer and just let it relax in space, it would look like ramen noodles boiled in water, just spread out. The minute that you introduce forced attraction, so forced returns, you can start to notice something really interesting happen, which is you start to form domain-like structures. The return process, though, is moving physical structures. You're moving nucleosomes onto each other. So they're going to interact with the volume that's present within the nucleus. And when you use that too simple system, which is that you have a property which forces returns, and the size of the monomers, you actually recapture very strongly the physical properties that you see on electron microscopy. You recapture packing domains. So now we have a system in place that's telling us why these things are forming in the first place. Knowing that returns would be anticipated to create domains, we then analyze molecules as the packing domains as a function of their size. So we organize them into small and poorly packed domains, large domains, large domains that are decaying, so poorly packed, and then small domain. And in RAD21 depleted cells, the mature structures are relatively stable. It's the nascent structures, the newly formed domains that aren't being made. So RAD21 is acting as an allocator. It's creating structures. What we can take from that is that TADs aren't necessarily a volumetric element. They're something else. And so what we now need to know is what are packing domains doing and how can we gain some information? So if cohesion is a system to define allocation, I need to have something that matures the domains and drives them into potentially functional units. It only generates half of the domains though. So we need to have another system to create allocations because even in the absence of cohesion, they still form. RNA polymerase II does that. So the act of transcription causes loops to form. And we can call those enhancer promoter loops or promoter loops or what have you. But the important part is that both CTCFs and cohesin create returns and transcription itself creates returns. So now we have two distinct modalities to cause the system to create nascent structures. To test whether or not transcription is acting as an allocation system, we used actinomycin D, and we did Hi-C and measured the change in connectivity, and you actually see a loss of loops when you do that. And the domains do something that we expected, which is you primarily lose nascent domains. With that, you actually gain decaying domains, so larger domains that are poorly packed start to increase in size, which is interesting on its own. So it would seem that transcription is involved in creating domains and potentially acting as a barrier element to their expansion. So I have an allocation system. So RNA polymerase II and cohesin act to define which segment should be grouped together. Now I need a maturation system. So I'm going to think about that. Going back to Myron et al., they proposed that the size of the molecules dictated how they would organize domains in space. So we tested that, and we tested that from the perspective of packing domains, which are mass fractals. Mass fractal is a really important component of the organization of the genome because it creates a continuous density gradient for function. You don't just have high density areas and low density areas that are independent of each other. You have a continuous unit that has a high density area, potentially an ideal area, and then an outer zone. We took SREV, which has the capacity, again, to capture domain structures observed on chrome stem tomography, and we introduced molecules that were roughly the size of a euchromatin molecule, a heterochromatin molecule, and a small molecule, and we looked at how they penetrate in space. And the way we achieved that is we took the configurations of these different protein classes generated from AlphaFold and calculated the radius of gyration. Euchromatin enzymes are about twice the radius of heterochromatin enzymes. So this potentially creates that hypothesized gradient. And when we run the simulations, that's exactly what we see. So you get a preferential positioning of larger euchromatin enzymes to the outer segment of a domain and a preferential deposition of heterochromatin enzymes into the higher density regions. So now we have a framework to say heterochromatin enzymes are going to be geometrically positioned based on the density gradient guided from domains. And euchromatin enzymes are going to end up on the outside. And then potentially something interesting could happen in the middle. To test that experimentally, we again went back to super resolution microscopy because it could give us molecular information. It could tell us how the different components of the genome interact with each other. We did 3-color imaging using H3K9 as a measure of high density heterochromatin areas, H3K27 acetylation as a measure of euchromatin, and RNA polymerase II as a measure of the active polymerase. And when we looked, you see this organizational principle. You see the heterochromatin organizes into a core, and around the periphery, you see euchromatin and active RNA polymerase. When you inhibited heterochromatin, or you inhibited transcriptional activity as shown on the bottom, you cause a dissociation between the domains and the active polymerase. And so that potentially indicated that heterochromatin was quite vital to the activity of the RNA polymerase far away. To test that, we then used chemical inhibition of a heterochromatin enzyme. We used a drug to inhibit HDAC3, and we measured the change in the properties of packing domains. So as expected, if the heterochromatin enzymes are necessary to mature domains, we would see an accumulation of nascent, so poorly packed small domains, and potentially small mature domains. That's exactly what we see experimentally. The things that decrease are the large structures and the large decaying structures. So there's a trade-off towards smaller domain volumes and an accumulation of nascent structures when you inhibit heterochromatin. So what does this mean? This means that you have a property of self-assembly. The act of generating strong returns, RNA polymerase II or cohesin, will create a nascent domain that has a gradient. That gradient then guides the positioning of the chromatin remodeling enzymes that then creates a mature structure that reinforces itself. If that signal goes away, the RNA polymerase II can still potentially transcribe around the surface, which we'll test next, while the chromatin remodeling enzymes provide a reinforcement system to keep those domains intact. And that leads to, is there a benefit of organizing our genomes in this way? There must be some reason that the system organizes in such a complicated manner. And the reason has to do with thinking about the RNA polymerase as a multi-subunit complex. RNA pol II is made up of around 12 different subunits and interacts with hundreds of different transcription factors. All of these have to diffuse to their targets, and then they have to remain stably bound. There is a benefit of local crowding, of essentially local mass, stabilizing the intermediate complexes. Basically, the entire system gains free energy from the RNA polymerase II remaining bound. But that benefit only goes so far. If the density gets too high, you start to impair the free diffusion of the reactant species, and so you get a peak. And that actually is seen experimentally from a paper concurrently with ours. When they added increasing crowders, they saw increased activity of RNA polymerase until the density gradient got too high. Okay, so returning to the concept of the mass fractal, well, we have a structure that's creating a continuous density gradient. And when you look carefully at that density gradient, you have an area that preferentially localizes heterochromatin enzymes, highlighted in red. You have an area that preferentially guides the transcription factors for binding sites. And then in the middle, you have an area that looks like the optimal zone for the RNA polymerase to do. We call it the ideal zone, or the habitable zone, if you will, of a domain system. And this is exactly what happens experimentally. When we look on live cell imaging with PWS, and we look at the positioning of RNA polymerase II, you get the deposition of the polymerase on the outside of packing domains. And the same thing was observed by Myron et al. So the domains themselves seem to be acting to organize transcription along an ideal reaction surface. This process completes the idea of self-assembly. The way to generate a transcriptional memory is the following. Strong transcriptional signals will form a nascent domain that creates a density gradient. That density gradient will then, guide the chromatin remodeling enzymes to create a mature domain of 3 components, and then the transcription will then occur at the outer boundary of that space in the optimal zone. We modeled this. So we took as an input what would happen if you have a strong signal causing returns to form on themselves. And you see a switch from the system essentially having entropic loops or random connections to force connections in a space. Over time, those force connections will accelerate a gradient such that you start to see accumulation of heterochromatin within the domain. And then the euchromatin fraction will go down, but what's really happening is your system is maturing. If you inhibited transcription, so if you added actinomycin D or you took away the strong signal, the domain won't form. So that coupled process means that I can take molecular information, pair it with physical information, and make a durable structure. It means, though, that you need heterochromatin, like vitally need heterochromatin to support transcriptional activity. So if you thought about cells that were in general around this ideal volume fraction, so this ideal level of heterochromatin within them, and you disrupted that process, so you went to a lower density state, you would actually see transcription turning off. We tested that. That's exactly what you see. So if you take an HDAC inhibitor, TSA, or an EZH2 inhibitor, GSK343, and you treat the cells with this, the heterochromatin amount decreases, and so does the amount of synthesized RNA. And this happens both near the periphery of the nucleus and within the interior. So the next question is, well, we can do this in an in vitro system. Is it true that the conjugate process is going to happen? Is the act of creating a transcriptional signature going to create a domain? In order to test that, we need to have a system that translates the 3D object back into the 1D object that's easier to study. The reason for that is, by and large, electron microscopy lacks the ability to tell us what particular transcription factors or polymerases are localized in space. So if you imagined that the RNA polymerase was moving in a one-dimensional line or an almost one-dimensional line on a reaction volume, what you would get is a projection in one dimension where you would have RNA polymerase would look like it's on the promoter and on the exons, and then potentially the heterochromatins depositing in the introns and the energetic space to create this schema. That's actually what happens. So if you take ChIP-seq and you look at the distribution of the active RNA polymerase and the number of active RNA polymerases on genes compared to how far away the nearest heterochromatin mark is, you see an inverse relationship. This is what you would expect if increased transcriptional activity at a surface is associated with a smaller domain versus a larger domain. Okay. Does this matter for physiologic processes? We therefore took muscle development because it's a well-organized system, and it could inform us about how the system translates transcriptional signals into the development of structural proteins like myosin heavy chains, which are quite large. This process in mammals involves the coordinated activity of 4 transcription factors, one of which is so potent it can transform fibroblasts into myotubes. So it's quite a robust system to test this idea of memory formation. So what do we see when we look at the data? The first is that in immature cells, in myoblasts, where there's low levels of transcription, there's a loop near the myosin heavy chains. It's consistent with rule 1, the idea that allocation is being created to create a gradient. When the system starts to amplify, if you look closely, you start to see deposition of heterochromatin in the intergenic and intronic spaces within the structural proteins involved. And then transcription benefits, sustaining this idea of a self-assembling system, guiding, using the RNA polymerase sequence specificity to guide the activity of the heterochromatin enzymes to then optimize the conditions for the process to take place. This looks like a reinforcement learning system. What does that mean? It means that if you take information about the state of the system, so what are the existing domains, what is the current nuclear volume, what are the ionic conditions in the redox state, and how many nucleosome remodelers that you have, and then you have as an input molecular signals, mechanical forces, and electric fields, the system is going to respond as a function of the existing domains, and it's going to iterate over time. And so you're going to have a way to guide what set of reactions you're going to have in the future based on the existing state. Does this matter for a disease process? Clinically, the loss of muscle has really negative outcomes. So independent of the cause of mortality, patients who are over 65 that have low amounts of muscle have a 2-time or higher all-cause of death. Domains are really sensitive to density and volume. And so from our modeling, what we see is that you will collapse domains if there's not enough volume to drive the formation of mature structures. But does this happen in vivo? And so experiments on muscle cells have shown that young muscle has far more heterochromatin, so far higher density compared to age muscle. And modeling from SREV, we can then generate domains and look at what happens to the behavior of the coding portions of the genes, the exons, relative to the actual structure that's at play. And we can do this because we can measure how many nucleosomes are in contact with each other. So that's the coordination number. More nucleosomes in contact, so more red, higher density, less nucleosomes in contact, blue. And the idea is that transcription is happening near that ideal zone. Somewhere between 6 and 7 nucleosomes is the ideal state. And as the density goes down, you actually slip away from the ideal conditions that you need to sustain transcription. But where are we putting heterochromatin to turn on gene transcription? That's antithetical to, at least 40 years of data, if not more. And the solution seems to be that there's segments of the genome that are not intended to be transcribed. And we refer to those as either introns when they're within genes or intergenic segments when they're between genes. And on the modeling, when you look at the density of the segments that are introns versus the density that are exons, it looks like actually the introns seem to have more crowding. They seem to have more packing in those segments, whereas the exons seem to be closer in density to the ideal zone, the positioning for reactions to happen. In the Myron et al. Paper, they propose something really clever, which is that the excess DNA helps to facilitate transcriptional activity or genome activity by controlling the accessibility of the genome. As I've just described to you, that process is actually guided by the activity of the RNA polymerases, by transcription itself. So that made us wonder, is the genome actually organized in such a way that in one dimension, you see echoes of the three-dimensional structure imprinted? And this would imply something pretty radical. The genome is actually organized around the act of synthesizing RNA and not necessarily on the replication of the genes themselves. So we went and looked for evidence of this process. So we took publicly available DNase, so measure of accessibility, And we looked at particular genes, especially large genes like myosin heavy chain 1. In addition to the accumulation of heterochromatin in the non-coding segments, you see a loss of access. And as I point out with the 2 arrows here, one is present within the intron and one is present within the energetic space. Is this actually happening across transcriptional activation? It is. We took the top 100 differentially activated genes, so genes being turned on in muscle development, and we measured what the accessibility is doing in the non-coding segments, so referred to colloquially by the term junk. And we compared that to transcription start sites. In the transcription start site, it's generally a stable configuration. You don't see a whole lot of change, but in the activation, in the developmental process of making muscle, genes are losing accessibility in their non-coding segments. Oh, sorry. This isn't restricted to in vitro systems. If you look at the heart, the brain, the epithelium, and you look at the patterns on ChIP-seq, you will see that there's heterochromatin depositing within introns and energetic spaces in highly active genes that are critical for the function of that cell. If we start to think about domains in the context of genes and vice versa, we can start to ask a question, well, maybe what we're looking at is how the genome is assembling individual genes and groups of genes in space so that they have the right configurations for them to happen. The length of genes is relatively easy to measure, and the length of domains and their composition is also measurable. And when you look at the histograms, compared to TADs, it actually looks like packing domains resemble the content of genes on the far left. You can convert by an approximation, so this is not the exact configuration that a gene adopts, a gene into a volume using the power law relationships. And when you do that, you actually see that many of our genes seem to be the right sizes to fold into packing domains. Okay, well, what does that mean? That means that every gene in our body has to almost carry enough non-coding DNA with it to generate the correct volumes. And it has to do it in such a way that's coordinated in the orientation of the transcriptional reading frame. I told you that you can take the length of the gene and estimate radius. And conceptually, then, you can ask the question, is the ratio of the amount of exon to the intron within the gene consistent with a surface area to volume relationship? A surface area to volume relationship, you would see that larger domains would have a smaller surface area to volume compared to smaller genes. So basically, you get an inverse relationship, which is what you see experimentally. That's lost if you randomly reposition the genes. You get a constant fraction of the genome as intron-exon ratios if you just randomly reposition the exons. And it's not just that there's a inverse relationship because genes are getting longer. What actually is happening is that the amount of surface that you're trying to create, so the amount of exon that you have, is dictating how much volumetric DNA is coming with it, so how much intron is coming with it. And that actually captures most of the protein coding genes and many of the non-protein coding genes. The benefit here is it gives you a mechanism to increase one-dimensional complexity. So you can store more information in one dimension because you can reduce it by creating a path along a volume in one dimension. So if you imagine genes in the orientation of the reading frame, walking along, skipping most of the introns as they fold in space, what you would see is that type of behavior. And to translate that, you have to look at a domain and calculate the fractional derivative as a function of the size. And when you do that, you see an inverse relationship between how well a domain is packed and the amount of volume that has to be carried with the gene. The fact that you can increase complexity in one dimensions means potentially that you could store a capacity to make complex states. That's kind of what you see when you look at the development of body plan complexity over time. So we pulled the genomes from yeast, C. elegans, zebrafish, flies, humans, and mice, and we looked at the surface area to volume relationship. In yeast, in baker's yeast, the genome is actually organized as a linear structure. It doesn't form into domains. And then gradually, as body plan complexity increases, you see a transition from a mix of linear plus domain-like structures to primarily domain-like structures. Is this process actually present across all of our chromosomes? To test that, we need two levels of information. One is to account for the fact that domains don't overlap in space. So you need to have a system that allows you to separate the domains from each other. And then it needs to be organized in the direction of the transcriptional reading frame. And I term that unit a hinge or a spacer. And what that contains is enough spacing DNA plus transcribable DNA such that it's about the size of a nucleus. And the reason for that is if it was too long of a chain, it would start to interact with the RNA polymerases in the manner that I was describing before that would generate domain-like structures. When you do that, the immediate consequences are chromosomes become organized as surface area to volume components. This isn't just because of the presence of exons. So if you take exons and you randomly reposition them, as shown in orange, you don't see structures that are domain length scale. You primarily see very large structures that are almost linear and then very small exons. When you move around the exon plus its adjacent non-coding segment, the domains will still form. You still have enough surface area to volume elements to create the right geometry. And this process seems to mirror the progression of body plan complexity. So the yeast genome, which is small, is actually quite linear. The amount of non-coding elements is proportional linearly to the amount of surface that you're trying to create. And as complexity increased, the elements started to require the formation of geometries. How does that relate to things that we typically measure all the time, things like connections and loops in space? So you can use a technique called chia-pet, which tells you when two specific proteins are in contact. And the more frequently they're in contact, the more durable you could imagine the structure is. So if you take loops that are formed by RNA polymerase II, which is the system that we are saying helps organize the genome, and you look at high frequency loops, and you look at their accessibility, you see a power law as a function of the accessibility. So the amount of surface area that's produced is proportional to the amount of volume that's encoded in that segment. On the flip side, if you look at loops that are not very strong, they actually have an inverse behavior. They actually look more like elements that are happening on the surface of domains. And so it's possible that you can actually separate out what type of structure you're seeing in that two-dimensional plane by accounting for a surface area to volume coupling in the genome. Why this is so powerful? If you start to think about these units as integrators of state with prior molecular signals, this process actually looks like a geometric computer. You take existing memories, you take new inputs, and then you get an output. And that means that cells in our body can shift their sizes, the amount of chromatin remodeling enzymes, the availability of ions, plus different molecular cues, and start to do geometric modulation to change the outputs. It doesn't require any mutational modification. With that, I'll take questions. The big implications here is it looks like our genome is a self-assembling system that creates several thousand packing domains that act as geometric processors inside of our cells. These domains are formed by the act of transcription. They look like transcriptional memories. And that this system was so powerful, it appears that it was imprinted onto the 1- and 2-dimensional structure of our genome as surface area to volume pairs. All right, I'll take any questions.
Boy, thank you so much. So let's take questions. If you have a question, you can just shout out, or you can put it in the chat, and we'll pick them up from there.
Luay.
Yeah.
Oh, perfect. I see also another question. May I go ahead and ask a question?
Yeah.
Thank you for this talk. It was amazing. Thank you. I'm a little confused with, there's so much information, but what happens with the loop? So how far away can you have genes come together to form a packing domain? I understand you're also talking about one gene alone forming a packing domain if it's large enough. But if we're, in the beginning of evolution, if you had smaller genes, or, how many genes could come together? And I guess the question I'm asking is, wouldn't that determine how the genes are packed in each chromosome? Because then you wouldn't have genes in opposing chromosomes coming together. So then is this determined by how much transcription is occurring? Could we figure out if they're co-transcribed genes, would they be similarly within same packing domains?
Yeah, those are all excellent questions. So the sort of the upper limit, I think, is encoded by what we see on chrome stem. So we see maybe the upper limit's like 2 million base pairs are going to form a single geometric unit. So that means that, if you have the average gene is around 2,000 base pairs. So you could be looking at maybe 10 genes at maximum working together in that volume. There is an important component that I think you really touched on, which is that the genes have to have complementary geometries. So when you make a packing domain and it's oriented in the orientation of the polymerase, the anti-parallel strand, if there's a gene there, will act like a volume element. So it may not actually get transcribed. It may actually get turned into a volume segment for the gene. And so you're actually going to start seeing what look like conditional patterns. So you're going to see logic-like behaviors in the system.
Very interesting. Thank you.
Yeah.
Thank you. And all right, so we'll take another question. It's a question from Ivan, and maybe I'll just read it, parts of it out loud. So the question concerns heterochromatic versus euchromatic factors, right? So you mentioned that euchromatic factors are larger, heterochromatic factors are smaller, and the question is that it seems like it does make sense that the larger things aren't going to be excluded from denser regions, right? But the smaller ones presumably would be everywhere. Would they be primarily localized within denser regions? Ivan, did I interpret your question correctly? Yeah, thank you. All right, so the question is, smaller heterochromatic factors, should they be everywhere equally, or primarily within denser cores of domains?
Yeah, it looks like it's more likely to be in denser domains. So the ratio for the larger things like the euchromatin stuff still actually favors the outside. It's just not as dramatic as the favoring of the heterochromatin enzymes to the inside. So there's like almost a 4-to-1 preference of penetration of a heterochromatin enzyme to a euchromatin enzyme to the interior. The inverse is there's like a 50% higher likelihood of a euchromatin enzyme to being on the outside, just from the model, without any other considerations like, the enzymes being multi-subunit or interacting with, other components and having like specific interactions with things like transcription factors. That's an excellent question.
But can you kind of like rationalize why small factors would be attracted to dense region? That's basically what the question is. Like it's not obvious why they should be there, why they should prefer more dense regions comparing to go everywhere.
Yeah, so it's not that they like prefer the dense regions, it's that their ability to penetrate the dense regions is higher. So all things being equal, if you had equal amounts of heterochromatin enzymes and euchromatin enzymes, and you looked inside of the domains as a function of their density, you would see more of the heterochromatin enzymes in the interior of the domain than the outside. But it's not like there's an attractive principle. It's not like they're looking for the high density areas.
And there's one more question, another question from Maria Unger. Does this mean that you can try to predict what genes work together just by their geometry? Maria, please feel free to elaborate or add to the question that I just read if you would like, but we can start with that. Right. Can you predict what genes work together just by their geometry?
I think so. I think that's there. I think that's what we're testing next. Not I think, we're testing that next. Because it provides like a very robust way. It provides a really robust way for your cells to trust each other. So if you were to think about cells during development all kind of getting similar signals, all of them kind of creating the same volume structures, and then when those signals start to dissipate, a non-specific system is in place to reinforce the process of maintaining them. All of a sudden, cells across an entire tissue have very similar domains. They have a very similar likelihood of responding in a coherent way with each other. Your question is asking, can I predict what the next response is going to be? And I think the answer is yes. So if we know what the existing domain configuration is, and you add a new signal, we should be able to say, here are the genes that should activate quickly. Here are the genes that are unlikely to work because of where they get positioned. And if I sustain the signal, and I was essentially telling the cells, this is what I want to encode, you're going to start to see new domains form.
Thank you for the question. So we are, let's see, right, so we are pretty much out of time here. Luay, I don't know if you could see the comments. Usually when I make a presentation using over Zoom, I don't have access to chat. So there are several more comments saying excellent talk, geometric code sounds exciting. So I think we are at the hour, so I think we can conclude that. There's one more question. Maybe we can take, okay, we can take one. Let's take one more question, yes.
Okay, thank you. So because it's so very interesting, and as Luisa said, it's very packed with information, so it's time to process. But one thing, of course, arises, like what happens between different cell cycles, right? So like your cell divides, you kind of like erase this dimensional information, and then new cell cycle begins, and what happens, like in your model, what happens?
Yeah, that's a fantastic question. So I think two things. I'm going to answer the inverse thing first. We have cells that don't divide, and those cells, if you look like neurons, heart, skeletal muscle, they're made out of big genes. Those domains are probably very, very hard to sustain in the right configuration through multiple cell cycles. So I think once they kind of get made, there's a reason why our cells like slow down. The second question, which is really quite beautiful about the system. So one, we didn't test it, but it's probably there. If we took the DNA methyltransferases and we started looking at how they started to get distributed in space, they probably will start to have a preferential positioning. And so there's a way to potentially modify the DNA so that the DNA is carrying a different state through the cell cycle division. That's one. The second one, which is the heterochromatin enzymes themselves start to make projections onto the DNA itself. So the cell is remembering what the last heterochromatin configuration was. So I think that's part of the solution. The last possibility is actually cohesin maybe helping to act like a chaperone, right? It's like, this was the prior allocation. When you go through your next cell cycle division, keep that allocation intact and keep memory. All of those are open questions. All of those are fantastic. So, you know, if you want to like work on that would be amazing.
Right. Why, thank you so much. And, you know, like I said, I'm a big fan of your work. And, you know, we heard some excitement from the questions and comments too. Thank you so much. If you have any follow-up, please feel free to send Luay an email. And I'm hoping to welcome all of you back in a month. Thank you.
2 weeks, I think.
2 weeks. All right. Sorry about that. 2 weeks. Yes.
I think it's Dr. Decker in 2 weeks.
Right. Fantastic. Thank you. Thank you.
All right, bye.